Okay, so I'm going to try to do one more video. I have a cold. I'm losing my voice, unfortunately. So far you've learned about in pregnancy, you've done fertilization, implantation, placentation, and so now we're at the end and we've got the fully formed fetus. I want to kind of go back and talk about some things that were involved in fetal development, genes, what gives you your hair color, and things like that. The first thing I want to remind you of, though, as we cover different units, anytime we have covered something previously that we kind of allude to in a current unit, you're still responsible for it. So I say that because we've already covered fetal circulation. So I just want to briefly remind you that you know this, okay? So it's still fair game. I don't have to go back and reteach it to you because you should remember it from circulation. But just in case you don't, I'll give you some kind of primers. If you remember, you have your baby inside the mom, and you have the umbilical cord, right? And the umbilical cord is going to then attach to the placenta, and the placenta is really some tissue from the mom and tissue from the baby, where the capillaries from the baby side are kind of bathed with blood from the mother side, which allows for gas exchange and nutrient exchange and things like that, right? We know that. So that hopefully will remind you that the fetus circulation is a little different. So if you remember, you've got to have a gas exchange in the placenta because the lungs are filled with water, amniotic fluid in the fetus, so they're not breathing for them, right? So we have to have gas exchange occurring at the placenta. So you have two sets of vessels that make up the umbilical cord, umbilical arteries and umbilical veins. So veins take blood into the heart, right? So this large red thing is the umbilical vein taking oxygenated blood, that's why it's red, into the inferior vena cava through the belly button. Okay, that blood travels to the heart, and then because the lungs aren't functioning, you send some blood to the lungs, but most of it is shunted by the ductus arteriosum from the pulmonary trunk to the aorta, or the foraminal valley from the right to the left atrium, correct? That blood can then travel to the organs, oxygenate those organs, and then the blood will return via the umbilical arteries out to the placenta. So again, I just want to make sure you remember that because you may see some questions regarding fetal circulation on your exam because we've covered it previously. So we want to carry that knowledge forward. We're not learning and dumping, we're carrying it forward. Okay, so let's get back to development. What makes boys, and girls, girls? And that is due to the chromosomes that you have. So what determines genetic sex? All right, so your biologic sex, what your chromosomes tell you. So remember, we have 46 sets of chromosomes, 23 pair. One pair are your sex chromosomes. You have X chromosomes and Y chromosomes. The X one's very large, the Y one's very small. So if you're female, you're XX. If you're male, you're XY. So each ovum always has an X chromosome, always. Males, the sperm, 50% of the sperm are X and 50% are Y. All right? So what does that mean? You have an X egg and an X sperm gives you a female. An X egg and a Y sperm gives you a male. That means your sex is determined by your dad, okay? So whichever sperm you get, X or Y, determines if you're male or female, okay? So your sex is determined by the male. And because the Y chromosome, the X chromosomes and Y chromosomes are different sizes, there has been people who've noticed that sometimes the sperm have different weights. And I know this is kind of weird. I just kind of want to talk about it just briefly because it's interesting. If you know anything about animal husbandry, about breeding animals, people have found that you can spin down the sperm and kind of separate out the X and the Y sperm. So when you're going to do in vitro fertilization or artificially inseminate a female animal, then you can kind of separate the X and the Y sperm and increase your chances of producing a male or a female. Doesn't always work because it doesn't always spin out just right, but there is some evidence and people know that if you can separate out the sperm, and make sure that, or try to make sure you get just females or just males in your offspring. So think about cattle. If you were trying to raise dairy cattle, would you want more males or females? You want more females. You want more female cows to make more milk. So in animal husbandry, they can try to increase the odds of having females or males depending on what they need, because the sperm are the determinants of being male or female. Now this is genetic sex. This is not the same thing as gender. Usually when we talk about gender, we're talking more psychologic sex, and so that's not really what we're talking about now. It's just genetic sex. Now what's important about that Y chromosome is it contains a specific gene called the SRY gene that's only on the Y chromosome, and the presence of that gene will initiate testis development and maleness. Okay, so you have to have that gene in order for the testes to develop and become male, phenotypically male. All right, so that reminds me, I said phenotypically, so what does that mean? Again, you've covered this before, so it's fair game now. You've got to remember alleles, homozygous, heterozygous, phenotype, genotype. Okay, you remember that, right? Yes. So remember, whenever you talk about some kind of a gene, we want to give it some kind of descriptor. So like brown hair, remember big B, little b. So big B's brown, little b's blonde, or let's do eye color. Big B's brown, little b is blue eyes, remember? So those represent the genes on the chromosome. Those are the alleles. They can be heterozygous or homozygous. If they're the same, they're homozygous. If they're different, they're heterozygous, right? You think about alleles, one's usually dominant. That means if you have that allele, you're going to have that trait, right? And one's usually recessive. You have to have two of those in order for the trait to show up. You guys remember this from blood typing, I know. All right? So remember, there's a difference between genotype and phenotype. The genotype is the genetic makeup. Big B, little b, big B, little b, little b. The phenotype is what you see, the expression. Okay, so for example, big B, little b are both brown, and little b would be blue. Okay? You also need to remember how to do a Punnett square. We've done that before too. So I just did a reminder here to show you how that looks. I did tongue rolling. I bet you didn't know tongue rolling was genetic. So that's tongue rolling. You can only do it if you have the gene. Okay? So here's mom and dad. They're both heterozygous tongue rollers. That means they both can tongue roll. So if they get together, you have these possibilities. Remember how you cross and do your multiplication and fill out your square. These two together, these two together, these two together, and these two together. So your genotype, you have 25% tongue rollers of TT, 50% big T, little t, and 25% little t, little t. What's the phenotype? 75% tongue rollers and 25% not tongue rollers. Okay? So just remember how to do that. I also want to make sure that you understand that when we talk about these traits, they're carried on genes. But they're carried on chromosomes, that genes are carried on chromosomes. But remember, chromosomes carry more than one gene. Okay, so I don't want you to get the idea that there's 46 chromosomes and only 46 genes. Each chromosome carries many, 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 many genes. And a lot of times, chromosomes will carry genes that we think about as being related. For example, you have blonde hair and blue eyes, for example. So hair color and eye color can be on the same chromosome. All right, so things that typically are expressed together because they're carried by the same chromosome. I'm really sorry, I've been sick all weekend. The other thing I want you to point out is I don't want you to get the idea that somehow the dominant gene is the good one and the recessive gene is the bad one. That's not necessarily the case. They're just dominant and recessive. It is totally possible that to have the recessive trait or the recessive genes gives you the desirable condition. So let's just do a couple of these so you can see the difference between something being desirable if it's dominant versus recessive. First one, the tongue rolling. Okay, if you have a dominant allele, so big T or big T little t, you're going to be a tongue roller. If you have the recessive gene, you're going to be a not tongue roller. You can't do that. Does it matter? No. So really, it doesn't really matter which one you get. Neither one's desirable. It just is what it is. But what about vision? If you have the dominant gene, you get astigmatism, which is a misshapen cornea. If you have the recessives, you have normal vision. So in this case, that's the desired trait. So having the two recessive genes, that's the good one. Let's look at this one. If you have the dominant trait, you have webbed digits. If you have two recessives, you have normal digits. So in this case, the recessive would be the preferred one. If you have the dominant for skin pigmentation, you have normal skin pigmentation. If you have the recessives, you have albinism. So in this case, you would probably say that the dominant is the preferred condition. I just don't want you to think that having the recessive is somehow bad. Sometimes it is what you want. Here's an important one, something called Huntington's disease, which is a genetic disease. Typically, it's fatal. If you have the dominant allele for that, you will get Huntington's. But if you have the recessives, you won't get Huntington's. So obviously that's the desired condition. So again, just with the understanding that recessives can be the desired condition. I also want you to understand that some things only come from mom. You only get them on the X chromosome. And so, well, they don't necessarily come just from mom. They only come from the X chromosome. So we call these sex-linked inheritances, okay? So these are X-linked genes. So they're found only on the X chromosome. So if you have an X chromosome and you pass that on, you're going to, with that allele on it, you're going to pass it on to your offspring. Now, this is important, especially when we think about males. So let me say why. So this is a female, right? And so let's say you have a disease, let's say it's hemophilia, and let's say mom has big H and little h, okay? That's what she is. That's her genotype. Her phenotype is that she does not have hemophilia. Let's say that big H is the good one, okay? So you don't have hemophilia. Now, let's say she now passes her genetics on to her offspring. Well, if she has a son, let's say she has two sons. And this son, he gets this one, and this son gets this one. This one is protected. No hemophilia. This one has hemophilia because there's nothing on the Y. The hemophilia gene is not carried on the Y chromosome. It's only from the X. So X-linked alleles, X-linked recessive alleles are always expressed in males. If she had daughters, and so it would not be expressed because they would have the dominant. So in males, the X-linked recessive allele is always expressed. And this means it's always passed from the mother to son. So for example, hemophilia or red-green color blindness. So guys, if you have color blindness, you got that from your mom, even though she may not be colorblind because she has a dominant allele to override that. All right, so the next thing we're going to talk about is development of the tissues and development of the sex organs. So the first thing is kind of to go back and think about what's happening after implantation. Those first divisions of the moella became the blastocyst, and you started seeing differentiation. So some of the first differentiation you see is a division into an ectoderm, which is outside, mesoderm, middle, endoderm, which is inside. So basically you're getting the early precursors to the digestive system, the bone, the muscle, the skin, and the nervous system. Okay, that's the first thing. You kind of divide the body into an inside, a middle, and an outside, and those are going to start going down a pathway to become certain organs. The other developmental thing that has to happen is it going to be a boy or girl? And so there is a stage that is sexually indifferent, where you have tissue that can either become a penis or a clitoris. There's tissue that can become a scrotum or a labia, and it will become female if there's no testosterone present. So having that SRY gene is important. The SRY gene not only initiates the production of the testes, it also turns on a gene that causes the production of testosterone. So if testosterone is present, this indifferent tissue will become male. So here's at 5 weeks. So in the presence of SRY and testosterone, you're going to get this glans penis forming a penis, a glans penis with a penis, and you're going to get a scrotum. Okay? With no SRY gene, that same indifferent tissue will then form the clitoris, the labia majora, the labia minora. Okay, so you can just follow the colors down. So the SRY gene and testosterone will cause that indifferent fetal tissue to become male. About 2 months before birth, you're going to have another testosterone. You'll be having more testosterone formed, and that testosterone is actually going to cause the testes to leave the body and go toward the scrotum. Because during development, the testes are actually formed up inside the abdominal pelvic cavity, and they have to leave the abdominal pelvic cavity, and descend. And they do that by following the gubernaculum. I kind of think of it as a zip line. So they grab hold of this fibrous cord and they zip on down through the inguinal canal out into the scrotum. So the gubernaculum will guide the descent. The ovaries descend a little bit, but they don't go very far. They just kind of go kind of parallel to the top of the fundus of the uterus. And they also descend a little bit, but not to the outside. So here is kind of the 6-week embryo in the indifferent stage. Again, we have the present of the SRY, which is going to be maleness. So you see the production of testes here, ovaries here with no SRY. At about 6 to 8 weeks, you're going to start to see the both gonads kind of start to bend and migrate. The ovaries are going to migrate to level the uterus, and the male testes are going to migrate outside. All right. So if a child is born with an undescended testicle, that means the testes did not leave the pelvic cavity and go into the scrotum. All right. All right. So just a few more things. Organogenesis. This is what happens, how certain tissues become certain organs. And so gastrulation is kind of sets the stage for organogenesis. And so somewhere around 8 weeks, you're going to start seeing all of the organ systems. Something is recognizable. It may not be fully functioning, but you can go, that's sort of heartish, liverish, lungish, kidneyish, okay? And once you get to that stage, you're no longer an embryo. You are now in the fetus. So this is somewhere between weeks 9 through 38. So that's probably that second trimester. Once you get to this period, everything's in place to start developing into its mature fetal structures and then neonatal structures. And this is a really, really fast time of growth. So what I want you to really take away from this slide, this is the last one I believe, is where your organ systems that you know about, where did they come from? So where were their germ tissues? So remember, you have your original embryonic cells that divide into those 3 regions, your ectoderm, your mesoderm, and your endoderm. And where do those go? What do those become? Well, the ectoderm outside becomes your epidermis and your hair and your skin and your glands of your skin, like sweat glands. It becomes the brain and spinal cord. Think about how superficial your brain and spinal cord are to the rest of your organs. Okay? You also get bone. You also get some of your sensory nerves and pigment cells and things like that. The mesoderm forms the notochord, and that's the your vertebral column. Okay, so it's going to kind of, well, not really your vertebral column, I guess your vertebral discs. Sorry, so the vertebral discs that go between your vertebral column. You're going to get the vertebrae here from the somites, are becoming your vertebrae in your ribs, some of your trunk and limb muscles. You're going to get what's called intermediate mesoderm, which becomes your kidneys and gonads. You're going to get lateral mesoderm, which is going to become your connective tissues, your bones and joints, your digestive system. So I don't want you to remember these intermediate stages. What I do want you to remember is ectoderm becomes this, mesoderm becomes all of these, and endoderm becomes your epithelial lining, glands, digestive and respiratory tracts. Okay? So again, don't stress about the middle stuff. Just kind of know the beginning and the end stages. All right, and if you think about what the words mean, ectoderm, mesoderm, endoderm, meaning outside, middle, and endoderm, that kind of allows you to know where it's going, what it's going to become. All right, that's enough. I'm about to lose my voice, so I'm going to stop here, and I will see you again in another video. Thanks.